Hello friends. So now in the last lecture we have done the types of muscles, particularly the striated muscles. We have also discussed the ultra structure and the fine structure of these muscles. Now we will be starting with the working of these muscles, how these muscles will be working, okay. So there was a scientist called known as H.E. Huxley. So Huxley and A.F. Huxley, these are the two scientists. So this both scientists gave an explanation of working of these muscles, how this muscle are getting contracted, and this theory is known as sliding theory, or we can say sliding filament theory. So it was given by Huxley. Huxley basically gave this theory by different experiments he conducted and the theories he made, basically how this will be functioning. It was observed that when the fibril contracts, contracts, its A band remains intact. We will see that in step-by-step manner. So whenever the contraction occurs, the A band, we have seen that in the striated muscles we have got two bands, that is A band and I band. A band was consisting the myosin, that is thicker, and the I band was consisting the actin filaments, which was comparatively thinner. Okay, now this A band will be remaining intact during the contraction. You have to note it down, it may be asked the question. So whenever this contraction will be taking place, the A band will retain its size. A band will not change its size. It will neither get shrink, it will neither get expanded. But during this contraction, the I band gets shortened. During this contraction, the I band will get shortened. Okay, and eventually it may get disappear as well. Or we can say it gets shortened by around 65% of its resting length. So when it is resting in condition, it's normal in condition. During that particular time, it will be at normal size. But when this contraction takes place, it will be reduced to 65%. 65% of its length will be reduced, and A band will be remaining same size. It will not be getting shortened. Okay, so here we can see. So this is this is the normal condition, and this is the contracted condition. This is the upper one is the normal condition, and the lower one is the contracted condition. So this is the A band. This is the A band. This whole red area you can see, this is the A band, which has got an M line passing through it, the center of it, and just surrounding this M line has got H zone. This is the H zone. Now and just toward the sides of this A band, we have got I bands here and here. And this from one Z line to another Z line, this whole structure is called one sarcomere. Okay. Now during the normal condition, this is the size of my A band. Okay. And this is the size of my I band. Now what happens during the contraction? So after this contraction also, you can see the A band size is not getting changed. The A band will be remaining same. But the I band you can see, it has got change in the size. Here you can see. Okay, so here you can see it has changed its size. The I band is basically shortened while the A band is same. Okay, so here you can see the A band is same while the I band has got shorter. So let us see. So here this is the A band. Okay, and here also the A band got the same size. This A band has got same size, but this is the I band. So this I band is basically shortened compared to there. It was a big compared here. It was a larger, but here it got shrinked. Okay, so that was about the A band and I band. Now let us see how it basically does happen. So sliding filament theory, where we start again. It is during the contraction, it was observed that the A band remained intact, as you have seen, while the B I band will be shortened by 65%. Now due to this, the H zone will also disappear. So because this is the H zone, you know what is H zone? This is the H zone around the M line. So what will happen due to this contraction? What will happen? This actin filament, this blue colored lines are basically mentioning the actin filaments are determining are basically showing the presence of actin filaments. So when they got contraction, when the contraction occurs, these actin filaments overlap each other. Here you can see they are overlapping. See, the gap was present, the H zone was present, but here when these actin filaments are coming near to each other, they will be coming that close that both the actin filaments of the adjacent I band will get overlapped. And because of this overlapping, you will not be able to see that light zone. That light zone was called as H zone. So here it will not be observed. So we can say that even the light H zone will be disappeared. So at this stage, the H zone also disappears because the actin filaments of both sides, we have seen that because the actin filaments of both sides in each sarcomere reach and may even overlap each other at the M line, and the Z line now touch the ends of the myosin filaments. So here we can see, so what happens, this is the relaxed state, normal state, resting stage. When it gets contracted, so the actin filaments will be getting towards the closer to each other, due to which the I band will be diminished or will be just getting so shortened, while the A band will remain same. But because the actin filaments are coming so close that they will be overlapping with each other, okay, and hence what will happen, the H zone will be disappeared. Okay, the H zone, that was a light band present between this A band, will be disappeared, and this Z line will be almost touching to this myosin filament. So here you can see, first there was a distance between there was a distance between Z line and this myosin filament of the A band, okay. But when they carry out the contraction, what will happen, this Z line will be shifted just besides to this myosin filament, and hence you can say they are almost touching this myosin filament, okay. So that was about the now during this condition, what happens, because the actin filaments of both sides in each sarcomere reach and may even overlap each other at the M line, and Z line now touch the ends of the myosin filaments, we have seen. And due to this, the size of the overall sacromere will decrease. So what is sacromere, we have seen? So this region between this Z line and the second Z line, so distance between the two adjacent Z line is called a sacromere. So here it was this was the length of sacromere. Now because of this contraction, the sacromeres, the Z lines came closer to each other, due to which the sacromere decreased. So keep in mind what all things we have to keep in mind. First of all, the A band will remain intact. The size of A band will not decrease, neither decrease nor increases. Second, the size of I band will be decreased. Third thing, the H zone will be disappeared. The H zone will be disappeared. H zone will be disappeared. Fourth one will be the Z line will touch the myosin fibrils. This Z line will touch the myosin fibrils. And the last one and most important is that during the contraction, the sacromere size will decrease. Okay. And all these phenomena occur in a reverse way to relax. Now, whatever things happen during this contraction, like the shrinkage of this I band and this disappearance of this H zone. So when the muscle will get relaxed, just opposite of all this phenomena will occur. So what will occur here? When it will relax, so this I band will get again increased. It will get increased its diameter. It will get increased. Okay, and H zone will appear again. You can write it down. So during relaxation, what will happen? During relaxation, the muscles comes in normal condition. It means the H zone will appear again, and the I zone will be increasing its size. Regaining its size. It will regain its size, and H zone will appear again, and the sacromere size will also regain. So these are the main three things which you will observe. Okay, and the actin filaments of the I bands will be moving far from each other. Okay, and even the Z line will be now moving far from the myosin filaments. Okay, these are all the things just opposite to the contraction will happen. Now the shortening of the fibril in contraction is brought about by sliding movement of the actin filaments. Now how this fibrils are getting contracted, or how these are getting shortened during this contraction, how these filaments are basically coming together and causing the contraction. So there's a phenomena for this we will be discussing. So shortening of fibril in contraction is brought about by sliding movement, and what type of movement it will be causing? Sliding movement, and hence it is called a sliding filament theory. Okay, so right now we are discussing about the main how this will be arranged. Okay, now let us see how it will carry out the sliding. So it will be, this contraction is brought about by sliding movement of the actin filaments over myosin filaments. So actin filaments will be sliding to the myosin filaments, and due to which it will be decreasing its size towards the M line by means of rapidly forming and breaking cross bridges or rachets at the spurs of myosin filaments. So we had seen the myosin filaments is having this cross spurs, we can say, or there is a rachets. Like, let us see, these are the this red color line is basically representing my myosin filaments, and the red color lines are basically having some sort of wedges present on them, you can see. So these wedges are representing the myosin head. We had seen globular heads were present, so they are representing the globular heads which will be binding to these actin filaments, we had seen the globular head were having the binding site of actin filaments. Here you can see again the globular head will be having actin binding site. Okay, so this myosin will be having this actin binding site, and this globular head which are projected by the cross arm are basically seen as the wedges over here, So what will happen during this contraction? This wedges or this globular head will come in contact or will bind with this actin filament at the active binding site, and they will carry out this contraction. Let us see how. So shortening of fibrils in contraction is brought about by sliding movement of actin filaments over myosin towards this M line by the means of rapidly forming and breaking cross bridges or rachets at the spurs of myosin filaments. These wedges are also called as spurs. Okay, now this will be this and this will be responsible for causing the contraction. So let us see how it actually happens. Okay, so let us see. It was again explained by the sliding filament theory, which states that the contraction, here you can see this in diagram first of all, then we'll see that thing. Okay, so here it is under the control of nervous system. You have to keep in mind it is under the control of nervous system. So the neurons will be responsible for secreting some specific chemicals which are responsible for causing the contraction. The neurons will be responsible for secreting some chemicals. Neurons will be secreting chemicals at these muscles, and this will be responsible for causing the contraction. These muscles will be the chemicals that are secreted is called as neurotransmitters. The chemicals that are secreted is called as neurotransmitter. What neurotransmitter it is? Acetylcholine. You can note it down. It is the acetylcholine. It will be the acetylcholine. So the acetylcholine will be secreted by the neuron at the muscles. Now this part where the neurons terminal, this is the neuron terminal, that is axon terminal, and the muscles and the muscle cell, there is this junction between the neuron terminal and the muscle cell is basically playing a very important role for this contraction and for this conveying this message to the cell. Okay, and this junction will be receiving these chemicals. This junction will receive the chemicals. What these chemicals are called? These are called as neurotransmitters. And neurotransmitters can be of different types, but right now here we can see acetylcholine, that is ACH. So this acetylcholine will be released, and this junction between this region between this terminal of neuron and this muscle is called as neuromuscular junction. So these muscles will be having some specific receptors which are called as motor receptors. These are the motor endplates which will be having ACH receptors, and according to these receptors the actions will be taking place inside the cells, mainly causing the contraction. Okay, so what happens when this acetylcholine is released at this motor end plate? It will be responsible for releasing the acetylcholine, and this acetylcholine will be absorbed or will be taken up by the receptors, which will signal the cell to open up the calcium channel from the endoplasmic reticulum. So when this acetylcholine enters into this cell, it will be responsible for increasing the calcium concentration. Why? Because this endoplasmic reticulum, or we can say in the case of muscles, In the case of muscles, this ER is called as sarcoplasmic reticulum. It's called as sarcoplasmic reticulum. So when the acetylcholine will be entering into the cell, this sarcoplasmic reticulum will open up and the calcium will be removing out. The calcium will come out into the cytoplasm. So when the acetylcholine gets into the cell, what will happen? The calcium channel present on the sarcoplasmic reticulum will open up, due to which the calcium will get entered into the cytoplasm. The calcium will get entered into the cytoplasm. Now this calcium binds to the troponin. We had seen troponin on the actin filaments. The troponins were the one which were responsible for masking the ATP binding site for myosin, you know. So we had seen that the troponin has responsible for masking, for covering the ATP binding site. They were responsible for covering the ATP binding site. Okay, so when these troponins are coming in contact with calcium, so calcium will bind to troponin. Calcium will bind to troponin, and due to this, they will be exposing the myosin binding sites. So when this calcium will bind to troponin, now this troponin is not able to cover this myosin binding site present on it. So it will not be able to cover the myosin binding site present on it. So it will be it will be active now, it will be free now. So this myosin binding site will be binding to the globular part or globular head of the actin. So this is the actin, this is the actin, and this is the myosin. Okay, so what will happen, this myosin will now connect to the globular part of the, this actin will be connecting to this globular part of the myosin. Okay, and this is caused due to the binding of calcium with the troponin. So when the calcium binds to troponin, the troponin will unmask, or it will uncover the ATP, it will uncover the binding site for the myosin, due to which now this myosin will bind to this actin. Myosin will bind to the actin. This binding will be associated with the ATP cleavage also. It will be associated with the ATP cleavage. So when this will be free, this ATP, we had seen that even the globular head of the myosin protein was having ATP binding site also, and it was also able to cleave the ATP because it was ATPase enzyme. It is an ATPase enzyme. So this ATP will be used over here. So this ATP will be providing energy for it. So ATP will be providing energy for it, for this particular, and hence this will be having high energy structure. So it will be responsible for formation of this complex between actin and myosin, and further it will get broken down and another complex will be again formed. So this cycle will be continued. So here what will happen, this complex is formed, and then once it is getting free, it will get contracted. Due to this, it will get contracted. So when this myosin binds to this actin, the actin will move forward direction. You can see this arrow represents it is moving in the forward direction. So it is moving in the forward direction, means it will be getting contracted. It will be getting contracted. Once the calcium is again low, so calcium will be getting inside this endoplasmic reticulum or sarcoplasmic reticulum. Now in the absence of calcium, again the troponin will be binding to the active site, and it will be preventing the binding of actin with the myosin, and hence the contraction will be not present. Okay, so let us see fatafat once again. So what happened? Mechanism of muscle contraction. So it was explained by sliding theory. Here why we call it sliding, because here you can see these actin filaments are getting slided. These actin filaments are getting slided. You can see this arrow are getting slided upon these myosin filaments, by the bonding, bond between them, by getting attached to it. Okay, so it was explained by sliding filament theory, which states that the contraction of muscle fiber take place by sliding of a thin filament over thick filament. Thin filament are actin filament, thick filament are the myosin filament. Now the muscle contraction is initiated by a single signal sent by a central nervous system, we had seen. So it will be carrying the nerve which will be having the acetylcholine neurotransmitter. A motor neuron, we have seen a motor neuron. A motor neuron is that neuron which is responsible for carrying out the movement of the muscles, So a motor neuron is the one which is responsible for carrying out the movement of a muscle. So a motor neuron along with the muscle fiber connected to its constituent, a motor unit, is called as motor unit. So this is the motor neuron, and this is the muscle fiber. Both together are called as motor unit. Okay, so a motor neuron along with the muscle fiber connected to it it constitutes a motor unit. The junction between a motor neuron and the sarcolemma is that membrane of the cell, membrane of the muscle cell. Okay, so the junction between the motor neuron and the sarcolemma of a muscle is called a neuromuscular junction or motor end plate. So here this is the junction between this nerve and this motor, this cell, sarcolemma we can say, and this region is called as neuromuscular junction. This region is called as the neuromuscular junction, or we can call it as a motor end plate. You can say this is a motor end plate, or we can say neuromuscular junction. Okay, so once it occurs, now what happens? A neural signal reaching this junction releases a neurotransmitter. What is neurotransmitter? The chemicals responsible for stimulating the impulse or generating impulse in the neuron. Now this neuron transmitter will be the acetylcholine over here, which generates an action potential in the sarcolemma of the cell. This spreads through the muscle fibers and causes the release of calcium ions in the sarcoplasm. Sarcoplasm is the cytoplasm of the muscle cell. So it will be responsible for releasing the calcium ion, and this calcium ion leads to the binding of calcium with a subunit of troponin, as we told that. So it will bind to the subunit of troponin on actin filament and thereby remove the masking of the active sites. Now the active site was basically covered by this troponin, but when this troponin binds to the calcium, it will not be able to bind the active site. So hence the active sites are open now. Now these active sites will be binding to the myosin globules. Utilizing the energy from ATP hydrolysis, as you see, the myosin globular head will be having ATPase enzyme also. So it will be binding to the myosin head now binds to the exposed active site. Why it is exposed? Because calcium is present. It will be binding the exposed active sites on active to form a cross bridge. So this here structure you can see here. This is the bridge structure present between the actin and myosin. This blue is the actin one, and this brown is the myosin. They both are connected with forming a bridge. It's called as cross bridge. It will be formed for a very short period of time, but during this cross bridge, what will happen? This actin filament will slide over this myosin, and hence they will get contracted, and hence they are called as sliding theory, and it is called as sliding theory. So it is clear from the above step that during shortening of the muscle, that is contraction, the I band gets reduced. We know that, whereas the A bands will be retaining its length. Okay. Now the myosin, which is releasing the ADP and PI, as we told that it will be having ATPase enzyme activity also. So it will be releasing the ADP and PI, goes back to release state, and new ATP binds to, and the cross bridge is broken. So when the ATP is broken down, ADP and PI, so this will lead to the having high energy to this myosin protein, due to which it will bond, it will attach with this actin protein and will form this bridge. It will form this bridge. Okay. Now what will happen? A new ATP will bind, and this cross bridge will be broken down. Then the ATP is again hydrolyzed. Once the ATP is bound, keep it in mind, either they can form a cross bridge, or they can hydrolyze the ATP. So when the cross bridge is there, the ATP is not attached. When the cross bridge is not there, ATP is attached. So once the ATP again get hydrolyzed, after hydrolysis, the myosin protein will get more excited, more energized, and hence will be responsible for formation of the cross bridge again, and this cycle will be continuously repeated, and hence this will be responsible for the continuous contraction and relaxation of the muscles. So the ATP is again hydrolyzed by the myosin head, and the cycle of cross bridge formation and breakage is repeated, causing further sliding. We have seen that. The process continues till the Ca2 ions are pumped back to the sarcoplasmic cisternae. So until unless this calcium ions which were influxed, or which were just came into sarcoplasm through the sarcoplasmic reticulum, will be basically, now this calcium ions will be getting lowered, or will get again back into they'll pump back into the sarcoplasmic reticulum. And due to this, what will happen is troponin will be again get masked the actin filament, active site of actin filament. This will be responsible for return of Z line, as we have seen, back to the original position, that is relaxation, and the reaction time of the fibers. The reaction time of fibers can vary in different muscles. So different type of muscles, the one which are working so faster, like cardiac muscles, will be having a different reaction time, while the muscles which are very slowly occurring muscles, like the other muscles or body muscles, they will be having slow movement. So over there the reaction time will be higher. So the reaction time of the fiber will be not similar in all the muscles. Keep it in mind. The reaction time of the fibers can vary in different muscles. It will be varying from muscle to muscle, and mainly depending on different factors. Okay. Now next is Next is type of fibers. The fibers which we have seen can be of two types. It can be the red muscle fiber, or it can be the white muscle fiber. It can be red muscle fiber, it can be the white muscle fiber. Okay, so what is red muscle fiber? So name itself suggests it is red in color. So this red color is basically due to the presence of hemoglobin. So if the hemoglobin is high, so it will be having rather than hemoglobin, we say it will be having myoglobin. Having the myo means it will be the type of globulin protein present in the muscle cells. Okay, so we will say that as a myoglobin. So this will be having high amount of mitochondria, and as they will be responsible for the aerobic muscles, they are called as aerobic muscles because these muscles will be responsible for carrying out the aerobic respiration, due to which they will be forming the ATP molecules and hence will be producing high amount of energy. So this was the white, and other one is the white muscle fibers. White muscle fibers are the one which has got less amount of myoglobin, or maybe the myoglobin is absent. So this condition is basically found in the muscle fiber, white muscle fiber, and they will be appearing pale yellow or whitish. Okay, mitochondria are also few in them, so they will be carrying out mostly the anaerobic respiration. Okay, and the amount of sarcoplasmic reticulum is high. They depend on the anaerobic, mostly they will be carrying out the anaerobic respiration for the formation of energy. Okay, so that was about the red muscle fiber and the white muscle fiber. Okay, so we have seen the how mechanism is there, how this contraction relaxation will occur. What is A band, what is Z band, what is A band, what is I band, what is Z line, what is M line, what is S zone, what is sacromere. So you should be knowing everything, and you should be able to explain it properly because it's so important phenomena. Okay, next is the visceral or smooth muscles, or we can say non-striated muscles. Visceral muscles, smooth muscles, or we can say non-striated muscles. Now visceral organs are the organs which are basically present in the body. So these muscles are the one which are responsible for carrying out the involuntary activity. These are responsible for carrying out the involuntary activity. These are called visceral or smooth muscles or non-striated muscles. Let us see why they are called smooth and why they are called non-striated. So these are smooth, plain, and non-striated, involuntary in nature. They are involuntary in nature. It means we cannot decide their movement. We cannot decide their movement whether it has to move or not. We cannot decide. It is not under our control of consciousness. It is not under our control. So it mainly occurs in the walls of hollow internal organs like the alimentary canal. Okay, like the reproductive tract as well. So these are the structures which are hollow in nature within it. Okay, so these are mainly found in the walls of the hollow internal organs like alimentary canal, gallbladder, respiratory tract, uterus, okay, urinary bladder, blood vessels also. So these all are the hollow organs, and the walls of these hollow organs will be containing this muscles. Okay, now these smooth muscles of skin dermis. The smooth muscles may be found on the skin as well sometime. The smooth muscles may be found in the skin dermis as well, and over there the smooth muscles of the skin dermis are called as erector pili muscles. They are called erector pili muscles. So write it down. The smooth muscles which are found on the skin dermis are called as erector pili muscles. Okay. Now these are associated with the hair roots and are responsible for flesh. So what are flesh? You would have experienced sometimes when you basically see a thriller movie or something like that. So your hair on the body start goosebumps. Okay, you call it a goosebumps. So your hair on the body starts getting erected. So that is called as flesh of hair. It's called as flesh of hair. Okay, hair flesh. Okay, so this is mainly due to the erector pili muscle. And they are mainly found in the hair roots. They are found in the hair roots, the part where the hair gets originated. Okay. Now these are also responsible for the erection of penis. They are also responsible for erection of penis and its limping. So erection of penis during the reproduction and its limping. It is also basically due to the muscles, particularly these muscles. Okay. Next, the structure. The smooth muscles fiber are unbranched. They are unbranched. They will not be having any branching, and they are basically spindle in shape. Okay, and uninucleated and has no sarcolemma. Okay, so Mostly, let us say this is the structure. So here we can say this is sarcoplasm is present within the cell. Nucleus is there, single nucleus, and branches are absent, and it has got the smooth pointed end at both the sides, the pointed terminals, and hence it is called as smooth muscle. Hence it is called as smooth muscle. Okay. Now contraction is slow comparatively. It has got slow contraction and involuntary under the control of autonomic nervous system. So here the contraction is comparatively slow, okay, and involuntary. It means it would not be all under control, like the movement of the alimentary canal, the peristalsis, the movement of your glands, okay, is not basically under your control. You cannot stop them. If you want to, you cannot stop the movement of your stomach, okay. So these are the involuntary movement. These are called the involuntary movement, and because this muscle will be responsible for causing this involuntary movement, hence it is called as involuntary muscle, okay. Now, and this movement will be controlled by the ANS directly, that is autonomic nervous system, okay. It will be controlled by the nervous system particularly, okay. Now functionally smooth muscles are of again two types. Functionally the smooth muscles can be of two types, okay. So it can be single unit smooth muscle, it can be single unit smooth muscle. These are the fibers which are composed of muscle fibers closely joined together. They will be closely joined together and will be working as a single unit. Example, urinary bladder, gastrointestinal tract, small arteries, veins. So we cannot basically differentiate the single smooth muscle particularly. They will be working together for carrying out the formation of single unit. Next is the multi-unit smooth muscle. Are the composed of more independent muscles. So they will be not present in a bunch or present together. They will be present in an independent different location, and they will be contract as separate unit. They will be contracting and they will be functioning as a totally separate unit. Example, hair root muscle, as we've seen, each of the hair will be having a muscle cell at its base. So here it will not be present along with the group of other cells, unlike the cells single unit driven, right? So example, hair root muscle, muscles of the wall of large blood vessels, ciliary muscles, muscles of Aries, etc. are the multi-unit smooth muscles. These are the type of multi-unit smooth muscles, okay. And the next one is the cardiac muscles. So these are the smooth muscles you can see. Here these are the having pointed ends. These are having pointed ends and uninucleus. So they will be having single nucleus, and they will be carrying out the involuntary actions, okay. And they will be having both the type of filament, thick filament and thin filament. We have seen thick filament is basically the myosin and the thin filament is the actin, okay. So when it is contracted, so we can see when it is contracted the size is decreased. Here we can see the size of the contracted muscle is decreased, and when it is relaxed, the size of muscle will be increased. Now you can see here, so here banding patterns are observed. So as you had seen in the striated muscles, the striated muscles were having A band and I band. Striated muscles were having A band and the I band. So unlike that, here you cannot see this A band and I band. So we can say they are non-striated muscles. They are non-striated muscles. Okay. So we can call them as non-striated muscles, or we can say visceral muscles, or we can say smooth muscles. Okay. Spindle in shape, having pointed ends, uninucleated. Okay. And mainly almost involuntary in nature. Last one is the cardiac muscles. On the basis of position, we have seen cardiac, but name itself suggests that it is basically a type of muscle which is found in the heart. It is found in the heart, and mainly in the heart wall. So wall of the large veins, just where it is also found in the walls of the large veins. Which veins? Just where this enters into the heart. So like the superior vena cava and the inferior vena cava, that part at which it will be entering just into the heart will be also made up of cardiac muscles. Now it is made up of cardiac muscle, and hence called as the heart wall. Heart wall is basically made up of cardiac muscle, and hence it is called as myocardium. Myo is basically the heart, and cardium means myo means muscle, and cardium means the heart wall. So this heart wall basically made up of cardiac muscles. Okay. Now structurally these muscles are resembling to the striated muscles. You will be able to see that they are almost similar to the striated muscles. It means they will be having striations. They will be having these bands, light bands and dark bands will be present within them. They will be having these bands. Striated muscles will be having bands, but it will not be functioning similar to this striated muscles in the case of we have seen. Okay, so they will be functioning almost independently. okay, and they will be consciously controlled by the brain. So when they are, we can say they are consciously controlled by the brain, it means they are functioning as involuntary. So cardiac muscles has got the properties of both voluntary muscles, that is the striated muscles, and the smooth muscles. Like what the properties of smooth muscle, that it is involuntary in nature. It is involuntary in nature. It will be functioning by itself. It is under the conscious control of the heart. While what is the function, what is the property like the striated muscle, that it will be also having the banding pattern present on it. It will be having also the banding present on it, okay. But apart from this, it will be having one more characteristic which is not found in both the smooth muscle, neither in the smooth muscle nor in the striated muscles. That is it is branched in nature. It is branched in nature, so it will be having branches, okay. It will be having branches, and mostly it is uninucleated and cylindrical nature. Mostly it is uninucleated and cylindrical nature, having the single central nucleus, okay. And let us see. So this is the cardiac muscle, we can see. So these are branch, you can see the branches over here. The muscles are often having this striations, this dark band and light band are present over here, and this is the intercalated disc. So this is the diagram of the cardiac muscles, okay. So branching are there, but it will be involuntary in nature, mainly found in the heart wall and the superior inferior vena cava, just near the heart, okay. That was about the different type of muscles present in the heart and the different type of body parts, okay. So we are done with the striated muscles, smooth muscles, and the cardiac muscles. In the next chapter we will start with the classification of the body muscles. We will be starting with the classification of the body muscle. Thank you. Have a nice day.